let's learn about protein synthesis. Now, protein synthesis is a process that takes place inside the cell. And inside the cell, what happens is the DNA is expressed. And in particular, it's the sequence of bases in the DNA that are responsible for the order of amino acids that come together to form a protein. Now, what's very, very particular here is the word sequence. It's the sequence of bases that determines the sequence of amino acids. Okay? Now, in protein synthesis, we have a location here. We are in the nucleus of the cell. And inside the nucleus, we have our genetic material. Also, in the nucleus, there's a substance, a bit like the cytoplasm is in the cell. There's another substance called the nucleoplasm, like the squidgy stuff inside the nucleus. So there's other things inside the nucleus. In particular, these bases. Now, this is a nucleotide. Okay, I've just drawn the nucleotide as the base letter, but I've just put next to it kind of a red part just to show the rest of the molecule. Okay, what this actually is would be your phosphate, your 5-carbon sugar, and then your base. I'm representing these nucleotides here, and I'm going to call them free floating nucleotides. And there's many of them in the nucleoplasm. They're there. Also in the nucleoplasm, we have some enzymes. Okay, so if we are making protein, then we need to access the sequence of bases in the DNA. So along comes an enzyme called DNA helicase. And what this DNA helicase does is it breaks the hydrogen bonds between the bases, just in that region of the gene that we want to express. So this could be the gene for insulin. Okay, this could be the gene. Remember, a gene is a section of DNA and exists in different alleles. This could be the gene from here to here, could be the gene for insulin. And we want to express that gene. So DNA helicase comes along and it breaks the hydrogen bonds between the bases of that gene, exposing the bases. So let's add some bases here. If this was an A, that would be a T. If that was a C, that would be a G. And we'll keep going. Now these bases are exposed. The hydrogen bonds have been broken. What then happens, and that's the result of the work of DNA helicase. So that's very A-level vocabulary now. We don't really need to know the enzyme at GCSE, but there is an enzyme that does it, so we may as well put it in there. What then happens is now these bases are exposed. So what will happen is the base that is complementary to the exposed base will position itself alongside the exposed base because they're complementary.
Yeah.
Okay, so next to this one here, which is a T, this will come along here like this, and I'll just do 3 as an example. Okay, so we'll get the complementary bases pairing up and then forming either 2 or 3 hydrogen bonds with the base that's exposed. Okay, we can probably do one more. Put that one here. And the same thing would happen on the other side. Okay, so the same thing would happen on this side as well. I'm only going to focus on one of the sides here. Now we've got our bases in the correct order. What we now need to do is get that information to the ribosome. So to get that information to the ribosome, first of all, we need to join it up. So along comes another enzyme called RNA polymerase. And what RNA polymerase does is it makes these nucleotides that are next to each other join together, making messenger RNA. This is messenger RNA.
RNA.
Yes, messenger RNA. Now what happens with this messenger RNA, it's got the sequence of bases that we need. We need to get that now to the ribosome. So what that messenger RNA does is it leaves the nucleus through a tiny gap in the nucleus cell membrane called a nuclear pore. So only small things can leave the nucleus. So the RNA is small enough, mRNA is small enough that it can leave the nucleus. Okay, and as it leaves the nucleus, it will go to the ribosome. So let's draw it in the ribosome now. And let's take with us the bases. So we'll go with A, C, G, T, and so on and so on. Whatever, I mean, that would have been a G, so we'll put a G there. So now our mRNA strand has left the nucleus, gone to the nuclear pore, got to the ribosome, which is in the cytoplasm. Now, the bases are read in groups of 3s. This is called a codon. So A, C, G is read as a base, sequence of 3. What we're now looking for is a molecule that's inside the cytoplasm, which is called tRNA, T for transfer RNA. Now the tRNA has on it 3 bases, but also an amino acid as well. And this sequence of 3 bases is called an anticodon and is attached to different amino acids. Okay. Now if we look at this codon here, it reads GCA. What's complementary to GCA would be C, G, T. So inside the cytoplasm, the tRNA molecule that actually has the anticodon that matches our codon positions itself like that. It will move to position. And the important thing is it's putting that amino acid in that place.
Yeah.
It's putting that amino acid in that place. Okay. And then another triplet codon. Let's just say this was A. With what would be complementary to that would be TCA. And that could be to amino acid number 6. Then that anticodon on the tRNA attached to a different amino acid would position that second amino acid in that place. It's a sequence. It's the order of the bases that gives us the order or the sequence of our amino acids. Now this process continues and we could have, and this process just carries on until such a time we have our amino acids next to each other. Now amino acids are the polymer of proteins. So now that we've got our amino acids in the place where we want them, another enzyme will come along and form a peptide bond between those amino acids in that order. And the tRNA molecule will detach itself and go off into the cytoplasm to go find another amino acid. The same will happen for this one. Same will happen for this one. And what you end up having is the amino acids in that particular sequence. The sequence then matches the DNA base sequence, and then that gives us our amino acids in the right order to then make our protein. So for example, if this was a gene for insulin, then this polypeptide chain, amino acids all together, would then fold together into its very unique 3D shape to make the protein. For example, in this case, we're talking about the hormone insulin. Then this insulin would be then released from the cell so it can go into the circulatory system and do its job. So for example, if it were to be insulin, then this would be the beta cell in the islets of the beta islet of Langerhans cell, or the beta cells of the pancreas. Okay, so that is protein synthesis. One more thing to add. Everything that happens inside the nucleus is called transcription. Okay, so the fact that DNA helicase breaks the hydrogen bonds, expose the bases, the free floating nucleotides form the mRNA, that whole process is called transcription. Everything that happens in the cytoplasm is called translation. So the codons being read in groups of 3s, the anticodon bringing the amino acid into the right place, and then the tRNA molecule moving away to go get another amino acid, and then the amino acids being joined up. That is called translation.
Okay.
Okay.
Do you know the carrier molecules, miss?
Yes, the tRNA.
Okay.